with the identification of cytochrome C, APAP1 and caspase-9, a picture was emerging of the molecular events that lead to the irreversible commitment of a mammalian cell to the apoptotic fate. Apoptotic stimuli lead to the activation of sensors, which results in the direct or indirect activation of the effectors of the BCL2 superfamily, BACs and BAC. Once activated, BACs and BAC cause a selective permeabilization of the outer mitochondrial membrane through a mechanism that is still unknown. This permeabilization allows cytochrome C release from the intermembrane space into the cytoplasm. The binding in the cytoplasm of cytochrome C to APAP1 monomers triggers apoptosome assembly. The assembled apoptosome facilitates the activation of the initiator caspase, caspase-9, which, once fully active, cleaves and activates the effector caspase, caspase-3. The activation of caspase-3 can be considered the point of no return, the event that irreversibly commits a cell to the apoptotic fate. Active caspases induce the processes that lead to molecular changes typical of apoptotic cells, such as PS externalization and DNA fragmentation, which altogether result in the dismantling of the cell and its engulfment and degradation. How conserved are these molecular steps in the worm in C. elegans? Let's look at the genetic pathways in worms and mammals and compare them. Then let's see what we know about each of the steps at the molecular level. And let's start at the bottom. In mammals, a cascade of caspases is activated in response to apoptotic stimuli. However, in C. elegans, it appears to be just one caspase, CET3, that is activated. Why a cascade of caspases in mammals, but not in C. elegans? A cascade of caspases may allow the amplification of the apoptotic signal, like a snowball effect. It may also allow for additional regulation. Since cells undergo apoptosis very fast in C. elegans, within less than an hour, compared to presumably many hours or even days in mammals, an amplification step and additional levels of regulation may not be necessary in C. elegans. But that's just a speculation. Let's move up one step. APAP1-like proteins are required for the activation of the first caspases in both pathways. And in both pathways, these proteins assemble into an apoptosome, which is required for the activation of the caspases. In one of the last sections, I told you about the structure of the mammalian apoptosome, as determined by cryo-electron microscopy of single apoptosome particles at a resolution of 12.8 angstrom. In 2010, Yigong Shi and co-workers published a crystal structure of the C. elegans apoptosome at a resolution of 3.55 angstrom. So with much better resolution and therefore more detailed structural information. Interestingly, the C. elegans apoptosome turns out to be octomeric. It is a circular structure with 8 rather than 7 spokes. Furthermore, rather than a flat, wheel-like form, as had been suggested by the structural studies on the mammalian apoptosome, the crystal structure of the C. elegans apoptosome suggests a funnel or cone-shaped form. And finally, rather than up to 8 molecules of pro-CET3, the C. elegans apoptosome appears to bind only 2 pro-CET3 molecules. This binding is mediated through the CAR domains present in both pro-CET3 and CET4. the two ProZ3 molecules bound per apoptosome presumably are forced to dimerize, which then leads to their cleavage in trans and their full activation. So far, everything seems to be quite conserved at the molecular level. However, moving even more upstream in the pathways, things start to become different. In the mammalian pathway, there is cytochrome C upstream of APAP1. However, there is no cytochrome C upstream of Z4 in the C. elegans pathway. The difference between the mammalian apoptosome and the C. elegans apoptosome is in the way their assemblies are regulated. Remember, the assembly of the mammalian apoptosome is dependent on cytochrome C binding to monomeric APAP1 in the cytoplasm. And the availability of cytochrome C in the cytoplasm is controlled by the BCL2 superfamily and the effectors BAX and BAC in particular. Therefore, in mammals, the BCL2 superfamily indirectly regulates apoptosome assembly by controlling cytochrome C release. However, the C. elegans apoptosome can assemble in the absence of cytochrome C. The crystal structure was generated just with Z4. Therefore, apoptosome assembly in C. elegans is not dependent on the availability of cytochrome C in the cytoplasm. How is it regulated then? And does the BCL2 superfamily play a role in it as well? Yes, the BCL2 superfamily does play a role, and it even plays a direct role. It turns out that in C. elegans, the assembly of the apoptosome is not actively induced in cells that are destined to die by apoptosis, but is actively blocked in healthy non-apoptotic cells. In those cells that are then destined to die, this block is relieved, allowing apoptosome assembly and ProZ3 activation. How is apoptosome assembly actively blocked in healthy non-apoptotic cells? It is blocked through a direct physical interaction between the guardian Z9 and Z4. In 1997, a number of investigators independently reported that Z9 and Z4 can interact. Therefore, in C. elegans, the BCL2 superfamily and Z9 in particular directly regulate apoptosome assembly. Interestingly, a few months after these findings were published, it was reported in two independent publications that, similar to what had been observed in C. elegans, mammalian BCL-XL and APAP1 physically interacted. The authors' conclusions were mainly based on observations made with tissue culture cells that overexpressed both BCL-XL and APAP1. So maybe there are two ways to trigger apoptosome assembly in mammals, a direct and an indirect way. No, there appears to be only the indirect way, mediated through cytochrome C release. A year after it was claimed that BCL-XL can interact with APAP1, 3 independent reports refuted this finding. And it is now generally accepted that what had been reported was an artifact of BCL-XL and APAP1 overexpression. If apoptosome assembly in C. elegans is blocked through the physical interaction between Z9 and Z4, how is the block relieved in cells that are destined to die? Again, the BCL2 superfamily plays an important role in that. Remember that Z9 can also interact with AGO1, the C. elegans sensor. The way we think the block on apoptosome assembly is relieved is the following. In the 131 cells that are destined to die during C. elegans development, the sensor AGO1 binds to Z9. AGO1 binding to Z9 induces a major conformational change in the Z9 protein. This conformational change leads to the loss of the Z4 binding surface in the Z9 protein and, consequently, the release of Z4 from the Z9 protein. Actually, Z9 binds to a dimer of Z4, and this dimer is released upon AGO1 binding to Z9. 4 Z4 dimers can then assemble into an octameric structure, the apoptosome. The model on how AGO1 binding to Z9 triggers apoptosome assembly is based on biochemical data obtained by a number of investigators, and also on structural data. Yigong Shi and co-workers published a series of papers in which they described the crystal structures of Z9 alone, Z9 bound to Z4, Z9 bound to a truncated AGO1 protein, and the Z4 apoptosome. Not too surprising, the structure of Z9 also revealed a fold very similar to that of other BCL2-like proteins. And having structures of Z9 with either Z4 or truncated AGO1 protein allowed to reveal the dramatic change in Z9's conformation that is triggered by AGO1 binding. Just to reiterate, an important difference between the mammalian pathway and the C. elegans pathway is the way that the assembly of the apoptosome is regulated. In mammals, apoptosome assembly is actively induced by cytochrome C release in those cells that are destined to die, and cytochrome C release is controlled by the BCL2 superfamily. In contrast, in C. elegans, apoptosome assembly is actively blocked by the guardian Z9 in healthy non-apoptotic cells, and this block is relieved by the sensor AGO1 in those cells that are destined to die. Does cytochrome C really have no role in the C. elegans pathway? So far, there is no evidence that it does. However, so far, there's also no convincing evidence that it doesn't. Mutations in cytochrome C have not come out of any of the C. elegans cell death screens. However, the 2 C. elegans cytochrome C genes appear to be essential for viability, and the C. elegans cell death screens performed so far required that the mutants be viable. Therefore, mutations in cytochrome C genes might have been missed. Also, there are 2 cytochrome C genes that may act redundantly. Again, one more reason why mutations in cytochrome C might not have come out of these genetic screens. Is cytochrome C released from the intermembrane space in the 131 cells that are programmed to die? We have tried to determine that, but ran into technical problems, which we have not yet solved. Therefore, at least as far as I know, it is still unknown whether cytochrome C is released in cells programmed to die in C. elegans. Fact is that the C. elegans Z4 protein is missing the WD40 repeat domain of APAF1, which mediates the interaction between APAF1 and cytochrome C. Furthermore, the C. elegans apoptosome can assemble in the absence of cytochrome C. And why should cytochrome C be released if it is not required for apoptosome assembly? While it is still unclear whether cytochrome C is released from the intermembrane space in C. elegans, there is some evidence that another pro-apoptotic factor might be released. This factor is the C. elegans homologue of AIF, the apoptosis-inducing factor. In mammals, AIF is released from the intermembrane space of mitochondria concomitantly with or shortly after cytochrome C. And Ding Xu and co-workers published that the C. elegans homologue of AIF can be released from mitochondria in response to AGO1 expression. This observation suggests that the outer mitochondrial membrane might become selectively permeabilized in C. elegans as well. Maybe not to release cytochrome C, but AIF and potentially other factors. If that was the case, who is responsible for this permeabilization? Effectors such as BAX? Remember, there is no BAX in C. elegans. But I also told you a few sections ago that in cells destined to die, SED9 adopts a pro-apoptotic function. And I hypothesized that SED9 bound to AGO1 may have a function that is like that of activated BAX in mammals. And therefore, I speculate that if outer mitochondrial membrane permeabilization occurs in C. elegans, it might be dependent on AGO1 and SED9. And this is something we should really test.